Kaplun from Variantics in Framingham, Massachusetts.
All right. Where is it? This one. Yeah, here it is. All right. I'm presenting today a whole genome pipeline that we have developed at Variantics to identify rare disease variants, not only. But let's start with just a short question to wake you up, guys, a little bit before the coffee break. So who out of you here thinks that whole genome sequencing will eventually be standard of care and everybody will actually do it, right? Any, most, I think more than half agrees. And next questions. Who thinks that it's actually now good time to do it? Yeah. All right. Less. I actually also don't think that now it's a good time to do it because my opinion, it's a good time to do it was yesterday. Yeah. And let me try to show you why I think so. So we at Variantics have developed a whole genome-based clinical test that it's fully validated for clinical use and it's out there for, I think, a couple of years already. We keep working on it and enhancing it. And it's mostly was designed for rare disease diagnostics. Why actually to do it right now? It's quite obvious. Again, it's a very famous graph of cost of sequencing and anywhere since, according to this plot, 2016 or so, it's basically no justification for doing anything else than whole genome as a first-line genetic test. Just the cost benefit does not provide you for doing something else. For example, exome does not provide you with much. The cost and time that all these diagnostic odysseys create to go with just a smaller test just do not justify to using them anymore. Why is that? Because from whole genome data, it's not just more costly and huge amount of data, but it also allows to detect multiple types of variants, not just exomes, and only also in exonic and coding regions is actually better because it gives better coverage, more uniform coverage. But it also allows you to detect structural variants, have all kinds of STRs, tandem repeats, you have mitochondrial variants, and you also go beyond exome, you go into regions that you don't normally sequence. Not that many pathogenic variants are out there in intergenic and intronic variants, but they are there, you know that, and many talks here are dedicated to such variants. All these are missed by exomes and by targeted panels. So this particular test that I'm talking about that we have developed, it actually is fully clinical certified by US authorities. We went with this test through clear certification, CAP proficiency, all state licenses, and it's quite a lot of paperwork, but not just paperwork. It actually requires very deep validation. So all these types of variants that we detect, we had to prove that we are actually detecting and reporting them up to standards of tests that are targeted, individual tests, that we are looking at targeted panel genes better or on par with targeted panel. We can do mitochondria better or on par with targeted mitochondrial test, and so on and so forth. So what I will show here is also examples of validations that we did for this particular reason. And let's start with just regular variants. If you go and the regular SNPs and small indels. So this is example of validation study that we did. These are 3 genome in a bottle samples. And you can see that you are getting as close as 100% sensitivity, specificity, positive predictive value, and accuracy. Just these are typical standard tests that on top of this, we did also repeatability, reproducibility, and so on. So we ran like a few tens of these over a period of time, different types of genome in a bottle samples. And just to show you example of benefits of it, this is a patient case that was shared by one of our customers, who it's a Genomet, it's a larger sequencing provider in Russia. And they are using our platform and our services for detection. So here is actually a pathogenic, non-pathogenic variant, which is intronic outside of any targeted panels and exons. Nothing more practically, but whole genome can detect this variant and provide answer to this particular patient. Mitochondrial variants. So mitochondrial variants, we claim that because it's PCR-free, no-enrichment technology in whole genome, you get around 2,000x coverage there, and it allows you to go to about 2% heteroplasmy. But why? How do we prove it? How do we show it? So to do that, we have actually created mixing and sequenced mixes of different proportions of also genome in the bottle and some control patients, 15 different concentrations just to show where and which percentage of the mix we are able to reliably detect. So again, this is data for validation. Short tandem repeats as well. This is our proprietary methodology that overlaps in a way in some known algorithms that allow detection of rather short expansions, but we went further than that, and we are quite efficiently are able to detect expansions of practically any size. You can see here another example of validation study for tandem repeats, and since we are detecting more than 20 different tandem repeats, we have to validate each of them that we detect with known patients that are verified with alternative technologies, and you see the actual numbers that we are getting, they are really close to what is determined by other technology. And I think we are actually more precise because most of the techniques for these STRs are not very precise. But we always, you see here the number, number of sample I just took out of this huge table that we have submitted as proof of our validation study as documentation. I just took some subset. We have more than 150 cases that we ran whole genome and verified that we are able to detect. And in every case, we were able at least to tell if it is a pathogenic repeat extension. I'm not talking about the exact number of repeats, if it matches or not, but it always tells you if it is within pathogenic range or not. And again, these are a few patient examples that we have. Structural variants, there are all types of structural variants. Of course, whole genome can detect them, and it has no limitation on size. Some people can try to detect structural variants with exons, doesn't work very well, and you need minimum 3 exons at least, you need training set, and so on and so forth. Whole genome, you don't have it. The only type that is tough to detect is actual unbalanced translocations, if they happen in kind of repetitive regions, so you then have good breakpoints. I did show this part at last year's meeting, and if you guys are interested in what more in detail of what we do for this detection, you can look up at proceedings paper that just came out thanks to organizers few days ago. But it's out there, so let me just go through this without, like, it's different types of signal, like split reads, discordant reads, you have depths as well, and there are different types of variants that different types of signal are taken in consideration together with supporting signal, like a ratio of heterozygous to homozygous alternate calls of SNPs and so on. So it's a lot of evidence that actually allows you to tell if the structural variant is real. No time for this, just time for some, again, real patient cases from like, literally last few days that we got. Again, from same customer for us, Genome, you see here, it's homozygous deletion, very small, of just a few thousand base pairs. No array, no other technology can detect this, and again provides answer to this patient. Duplications as well. In other patient case, duplications of less than 20,000, and for most of arrays, reliable detection of duplications will be close to 100,000 or so. This is really just a couple of exons that you can detect here. Insertions, so insertions, that's how typical insertion looks, and IGV, okay, quite interesting. Don't have many patient cases, but this is actually a recent addition to the pipeline that is not in the paper, so just half a minute I'll talk about this. This is the detection of transposable elements. This one, I know maybe some of you will recognize this is allo insertion. You can say, you can tell it because it has this red poly A tail on one side in both cases. So it's important to detect this because right now, according to current statistics, is more or less between 0.5% to 0.8%, according to different studies, of all pathogenic variants that are found in rare diseases are actually caused by transposable elements. So that's, in big scale of things, it's probably not huge, but it adds answer to 5 to 8 out of 1,000 patients, which is, these are human lives. So it's important to detect, and nice addition to the pipeline. Let's skip this part. No time for this. Different types of annotations and filtrations by population frequency. All these supporting parameters that, again, if you are interested, you can look at the paper to justify. Some of them are quite cool. So, for example, how to tell real duplication from something that is kind of bias of variant calling is that over real duplication, you'll see change in allele ratios here. So you can see that here, for example, you have in this SNP, you have twice as much of one allele compared to another, and here as well. So you can see that here it's actually the heterozygous allele fraction is unbalanced. And again, it gets into consideration when the system is calculating and deciding if the variant is actually real or not. So here you see this is validation study that we did for structural variants in particular. And for structural variants, the minus of it that you don't really have very good true set. And even in the true set that exists currently with current genome in the bottle true set, we are getting to close to 80% detection rate of structural variants of any size. And again, for example, below 1,000 base pairs, nothing else can detect them. But when we're looking into, you can say 80% is actually not really high number, but when you're looking at clinically relevant cases, this percentage is actually much higher, because in clinically relevant cases, they are rarely happen. Vast majority of structural variants, general structural variants, they happen in kind of repetitive regions, in intergenic regions, where we have rearrangements of genetic materials, quite common. But clinically relevant, they are more, they produce more signal just because they happen in important regions where it's a little more easier to detect. And to show this, we actually did clinical validation with 60 patients with known causative structural variants, and we detected all but one. So it's like very, very good rate. And couple recent interesting kind of additions to diagnostic procedure that we have. So one interesting problem is detection of horizontal variance. So when you have some kind of indel, typically variant callers, they will call parts of it separately. They can just take this indel and divide it into 2-3 independent variants. And the effect of this independent variance not necessarily same as the whole thing. So we actually combine them into one, but we also provide these independent parts as well. And often annotation and effect is very different of the whole thing compared to the parts that you see. Compound heterozygous variants. That's also important. So again, we have automation in the system that allows to clinicians that use it to see the compound variants of small sequence change with structural variants. So here is a patient case with glycogen storage disease that has a compound of point mutation with 8 kilobase deletion. And that's what you see. It's basically striking. So you don't have to spend these hours to actually go through cases. It just shows you just a subset of very specific variants that you can look and easily classify. This is my last slide, so not take too much time. This is also whole genome based. What I can say, the analysis for whole genome-based analysis for rare disease diagnostics can actually start not with newborn, but it can start before the child is born. Because if the child already born with rare genetic disease, it doesn't matter if you put sticker on him. Yes, it can change your expectations. It can change in some case the treatment, but it will not change the fact that you have a child and family that is suffering. So we actually created a whole genome-based carrier screening test that looks into all conditions, not just 1 or 2 or 5 genes that typical screening tests look, and it looks into everything. And if you have potential of having a child with rare recessive or dominant disorder, you will actually detect it and you can take precautions. And it also allows it also benefits parents because it detects late onset diseases, anything from cancer predisposition and cardiomyopathies to Huntington and so on. Or for example, it shows if it is a potential of expanding FMR or Fragile X, things like that. So it's an ultimate test. And again, the cost-wise, it's not much higher compared to small panels that are just looking into very particular spots. That's pretty much it. Have any questions? Don't think we have much time for them. But, right.
You have the whole coffee session.
Yeah. Yeah.
questions that we have now? No? Everyone? No?
Yeah, I have a curiosity. So, which coverage would you recommend for basically running the test and making all the algorithms work? Which depth, average depth, are you kind of sequencing to be able to achieve good precision in your calls?
Yeah, so we are we have validated everything again. Since we had to go through validation, we went with very particular settings. So it was Illumina sequencing, 30x coverage, and these are the results that we are getting. I don't recommend going below 30x. And what you can go, for example, with BGI sequencing, which has slightly low sensitivity for STRs, but pretty much everything else is more or less on par, but it's still substantially cheaper now. In terms of coverage, we see that at 30x, if you elevate coverage a little bit to maybe 45 to 50x, you have slightly better sensitivity and specificity for small indels between 5 and 15 base pairs. Other than that, not much changes. So 30x is cost versus the benefit is optimum right now.
Yeah, my question is because I heard like for some people doing exome-based diagnosis that somewhere they require like 100x. Which I think doesn't make that much sense.
In exomes, you actually need this because you don't have uniform coverage. So to make sure everything is adequately covered, you actually have to go to higher numbers of coverage. But in whole genome, since you do have 30x on average, but you have very few regions that are covered below your detection rate. It's PCR-free, no amplification bias. It's really uniform and very well covered. So typically, we have 0.3% covered below detection rate, so it's very, very low numbers. Yeah. Yep.
Hi, it's interesting. I just have a question regarding screening of a healthy population. Mm-hmm. Wouldn't you expect to find a lot of disease increasing, risk increase in a lot of diseases and areas false positives?
Absolutely. So if you are looking into GWAS studies, you will see all of these in whole genome because GWAS studies by design, they look into common SNPs, right? But we are not focusing on these, we are focusing on actual pathogenic variants. So it was a paper published years ago in Science that they were saying everybody has like 50 pathogenic variants, and you have maybe even more if you look more broadly. That's not what we see. Like, we typically see maybe 1 or 2 truly pathogenic variants. You have, of course, more carrier risk. So you have, on average, you can have 5 to 10 variants that are carrier risk, that you have heterozygous form. But your spouse should have variants in same genes in order to get the child with this recessive condition, so it's not that dangerous unless it is some common allele in certain population, and that's where you can respond to it. We find some latent onset things, again, in cancer predispositions, we find quite often and report them as well. We find sometimes things that are hard to tell to people. We had case with Huntington, for example. People came with family planning, and it's not very good news for family. So it is a dilemma how to report and deal with such things. But typically, the reports that we deliver are not overwhelming in terms of amount of data.
I have two questions. The first one is what's the ideal source of DNA for sequencing? Either is a buccal swab or blood source, or and the second.
Just to quickly answer you. So one difference that we use from typical DNA and typical library preparation process for our technique is use insert size of 550 bases. So you need a larger amount of DNA. We use standard Agilent TrueSec kit. They come with two varieties: 350 that everybody uses and 550 that we use because it gives you better detection of STRs and structural variants. It's just larger piece of DNA. It's easier to map and with better reliability, so it gives you better signal for breakpoints and for repeat extensions that are larger. You need 2 micrograms of DNA. You can't get 2 micrograms typically from buccal swab, but you can get saliva or blood. Blood is a little bit better in terms of just the quality of DNA because it doesn't carry any like hamburger that you ate yesterday or bacteria. But saliva, you can potentially look into microbiome, for example.
And the follow-up question is, if I want to get my DNA sequence, do I need a prescription from my doctor, or I can
Right, yeah, so we are not direct to consumer. We are not 23andMe. Their business is in selling your data to pharma companies. They actually sequence, they lose money on sequencing and delivering these results and keep the data. Just to send you this box, it costs them more than what you pay for typically for the test. But we actually are clinical diagnostic labs, so you need a prescription for test, but any clinician, any physician, your primary care physician can prescribe like any other lab test, like like sugar in blood, right?
Okay,
Right.
so I think maybe we can let Alex go get some coffee.
Thank you.
If you're interested in talking to him some more, which I would be, please find him. Thank you, and we'll
Thank
see
you.
you after the break.